Great. So
Okay.
thanks everyone. It's a pleasure to be here and thanks for joining me late, well, late for some of us on a Friday. Um, so basically in my lab we're interested in the big kind of question of how tissue size and shape is controlled and how we can get this, really beautiful diversity in nature, even though at the very basic, level we're made up of very similar building blocks. And so the main focus of my lab is really to look at this from the role of mechanical forces. Of course, genes are important. So we study, for example, how gene expression and proteins and molecular interactions give rise to the different mechanical properties of cells and tissues, how they generate forces, and in turn, how this lead to the sculpting of cells and tissues into their complex 3D and even 2D shape. And then, of course, ultimately, how this all feeds back, onto each other. For example, how do the forces and geometry impact gene expression and molecular interactions? And there's this constant interplay, of course, that's important for driving the development and repair of tissues and organs. And in the past, we focused mostly on intrinsic properties, i.e., how are these forces generated within the organism and the impact on tissue shape. But in the past, sort of few years, we've become more interested in the role of extrinsic forces. For example, forces from outside of the cells, such as from the basement membrane, which is a type of extracellular matrix that's just outside of most epithelial and endothelial cells, but also actually forces completely outside of the animal. From the environment. And so today I want to share these two new stories, which are all unpublished. So just to, hopefully get your feedback on this. And to put things into a bit more context, we're interested in this really at mostly at the multicellular tissue level. All right. And generally we sort of split this conceptually into three stages. So one is how do the forces and molecules interact during development to get tissues into shape, but also then after development, how are tissues maintained in their ideal shape for the rest of the lifetime of the organism, which of course is a critical problem, which I think we often ignore, but that's actually key for disease or lack of disease, hopefully, right? And then finally, if we do get damaged, for example, injury, how do our tissues actually repair back into shape? But because of limits of time, I'll focus only on the first two aspects today. And if you want to talk about tissue repair, which is another major angle in the lab, I'm happy to discuss this separately another time. And so, in terms of this, in terms of in development homeostasis, we mostly use the Drosophila as a model system, in particular the wing imaginal disc. So here, for those who don't know, the wing disc develops over the course of about 4 or 5 days, and is essentially a single layer epithelium. So this would be what I call the XY or planar view, top-down view. And if you were to cut a line through the middle, this would be the cross-sectional view. Okay, and it's a fairly simple model for epithelium because it's essentially a monolayer, although it's a bit more complicated than that, but for the purpose of today's talk, it's think of it basically as a monolayer epithelium, and it grows and divides and grows and divides very quickly over, as I said, a few days, and it starts to develop some complex structures such as these folds, which is also kind of interesting if you want to understand 3D morphogenesis. But what's important is this development is incredibly robust. You can almost age developmentally the wing disc depending on the morphological features, and the final shape and size of the wing in a wild type animal is almost always identical. So development, as the nature designed it, is incredibly robust, and hence is a great system to understand control of development and how do we get this robustness despite fluctuations in environmental forces. All right. So in the past, we've actually developed different types of computational models. For example, this one, which is a finite element model to look at wing disk folding. So again, this was published quite a few years ago, so I don't want to go into the details of this in today's talk. But since then, we've also been expanding the model to model different sort of tissues. So for example, we can model spherical geometries to start with, but then also how do you have symmetry breaking events, for example, and to answer the questions of, for example, how do you actually have differential growth, maybe coupling with differential mechanical stiffness or mechanical properties or viscosity and so on, and how do you generate patterns in kind of this interaction between the cells and the immediate environment. So again, these are things we are working on. But again, I don't want to focus on this today. Just this is just to show you if anyone wants to talk about this, happy to discuss. But what I would like to focus on today is, as I said, the problem of tissue. Sorry, I'm trying to move the slides. Oh yeah, oh, tissue homeostasis. So how do we actually stay in shape, right? So most of us, I mean, you're all probably sitting down. I don't know, some of you could be running while you're listening to this talk. But our organs are actually still moving and actually all at different speeds and rates and peristalsis, for example, of the gut after a meal, or if you go for a run, your heart's beating at different rates, right? So these, what I call fluctuating or variable or stochastic forces, we're always experiencing in our lives. Yet we don't constantly change the shape of our organs, right? Despite these fluctuating forces. But we know our organs respond to forces. So there's kind of a bit of a conundrum here is, how do we buffer some forces, but also allow some forces to be instructive and induce shape changes? And so I guess the question is, presumably it's to do with the nature of these forces, right? And so what I call therefore normal but stochastic or variable forces, we can tolerate them. Our organs are able to somehow buffer them, at least after, development and keep in shape. But of course, there are also extreme cases where too much force can generate, for example, in this case, heart hypertrophy. So clearly our cells and organs are mechanosensitive. And if you have too much, it can be pathological and it can lead to diseases such as hypertrophic organs and so on. And so the question there is, how do we, first of all, what is normal regime of forces? And is this different for different people, different animals? And also, how do we buffer these then? And then how much is too much? What is the limit of extreme forces and what are the consequences of them? I guess, hopefully one day we can manage them, maybe differently for different people, but we will have to understand these problems, right? And so to try to answer some of these questions, what we've had to do in the past, many years is to develop tools where we can apply controlled external forces to our tissues of interest and then monitor the consequences of these. And when I mean consequences, there are different ways to assay that, and I'll go into that. So, for example, this is a typical device that we use for ex vivo culture of organs. So we cut out our favorite tissue, the wing disc, from the animal larvae of the fly, and we can culture this tissue inside this device. So for simplicity, basically we clamp them between two layers of PDMS. This allows the tissue to be clamped on either side and holds it steady like two little, tweezers. But then there are also channels in the middle here and that where we can perfuse growth medium, which of course is essential for the organ to grow in the dish outside of the animal. And we can do this for about 5 hours without too much of a problem for the tissue. And then this whole device we can put into this kind of double bar stretcher device. And then the two bars, we can either expand them by rotating this kind of motor, or we can also contract them. So this is bidirectional. We can either expand or contract, okay? And so this allows us basically to apply, for now it's uniaxial, but we can either stretch the tissue or compress the tissue. We can also have the motor working at different speeds and has different cycles. So you can design essentially different frequencies and magnitudes of forces that you're applying, or strains, let's say, that you're applying to your whatever organ of interest, okay? And so for today, I will focus just on a single, pretty much immediate stretch and tell you a story about that. But in the future, we can, as I said, look at different cycles and so on, different regimes. And so this is just to show you a typical example of the kind of data we can get. Oh yeah, sorry, what I forgot to say is what's cool about this device is that we can put this on top of a microscope and do live imaging as we are perturbing the tissue. So almost all the data we do is performed through live imaging so that we can look at immediate live responses of cells and tissues to these perturbations that we're applying. Okay, and so this is an example of where we have the wing disc here is a top-down view, and this is a control anchor. So we always compare to the control where this tissue is in the device, but it's not being stretched. And we only look at the middle. Okay, imaging is only for the middle. These sides we ignore because they're also compressed from the top bottom. And hopefully you can appreciate that when we apply the stretcher, this middle region of the tissue is expanding, right, in the X direction, X axis. Okay. And One thing we observed is that actually within 2, 3 minutes, the actomyosin cytoskeleton immediately repolarizes upon this force application. So here you see myosin 2 in cherry, and essentially after a few minutes, we see it polarizing along the axis of stretch. And this is not just an optical illusion because if you look at E-cadherin, which labels the basically junctions or outline of every cell, this does not polarize. And you can still see these vertical junctions, whereas with actin and myosin, you can't. Okay. And so again, this is published a while ago, but the long story short here is just to show you that essentially we found one way at least to buffer these transient forces. So if you were to stretch a tissue, within minutes, actomyosin will polarize. This actually is via diaphanous, a formin pathway. And this polarization essentially stiffens this tissue so that it can no longer be extended as effectively. And this prevents the organ from being deformed during, for example, fluctuating forces in the animal. And we show that if you don't have this molecular pathway or mechanosensitive pathway working, then the wing actually develops into a malformed shape. Okay, and so this is one way, for example, in which you can change your mechanical properties very quickly, stiffen up the tissue so that you prevent permanent deformation and of these transient forces, right? But this only seems to work on kind of quite short time scales, as I said, minutes of fluctuations. But what about longer term deformations? And maybe you also still want to kind of buffer these, right? And so when we did this work, we realized that actually if we apply the stretch only for about half an hour, 15 minutes, and you let go of the stretch, everything is reversible. The cell shapes go back to the original, so it's purely elastic within this half an hour regime. Okay. However, we also noticed that if you were to maintain the stretch for, let's say, 3, 4 hours or so, and then you let go of the stretch, then actually the cells are permanently deformed. They do not return to their original shape. Okay. And so this shows that the tissue actually has undergone irreversible deformation or plastic deformation. But this actually, what we call this mechanical memory, seems to last much longer actually than what others have noticed. So, for example, we assume that within this at least 60 minutes or so, the memory is still there because if you let go, then it goes back to normal. But only after about 3, 4 hours of stretch, if you let go, then it doesn't go back to normal. Okay. However, if people have shown that in, let's say, MDCK or other epithelial cells where you just have a single layer of cells, then if you were to stretch them, even after about 2 minutes or so, if you let go of the stretch, actually the whole tissue starts to buckle or fold because actually that means the cells have undergone irreversible extension. And if you let go of the stretch, then the only way, of course, to fit that longer tissue is to fold the tissue right into this device. And so this is a much faster time scale. So here, essentially, what people have shown is that if you have just actomyosin-based remodeling of cells, this can occur within minutes of time scale. And people have done FRAP and so on to look at actin turnover, and that fits with the time scale. However, in the wing disc, it seems like this time scale of stress relaxation takes much longer to occur. And this essentially maintains the elasticity of the tissue for longer than if it were just cells. And so we asked ourselves, what is the difference between this epithelium and other simple epithelium? And so, of course, this wing disc has more than just cells in the epithelium. So, as I said at the beginning, effectively it's a monolayer, but actually I lied to you because also around the whole tissue and basal to this monolayer cell. So if you look at this tall cell, these tall cells, very tightly pseudostratified and packed, at the basal side of these cells, you actually have basement membrane. And it's been shown that if you remove the basement membrane with collagenase treatment, so the yellow basement membrane is gone, then the tissue actually loses its shape. So the basement membrane has a huge impact on the shaping and maintenance of the shape of the wing disc and in many other organs too, but the wing disc is a model system to look at that. And so then this is why we got interested in the basement membrane to ask, well, how does the basement membrane actually maintain tissue shape in animal systems? And so just to therefore explain some of the next set of experiments. So essentially what we want to ask is the stress relaxation of the tissue, which takes basically 3 or 4 hours to relax the stress, right? Why does that take so long? Is it because there is the existence of a basement membrane, which we hypothesize relaxes and remodels much slower than just cells and the cytoskeleton? And as a result, this is what maintains the memory or maintains the stress in the system, which is what allows the mechanical memory to persist. Okay, so hopefully that's clear. But how do we assess stress relaxation? And so the way we measure stress relaxation or the existence of stress actually is to do this experiment called an annular laser ablation. So essentially what we do is we cut a donut and which leaves a middle island and also an outer kind of ring, okay? And if the stress is isotropic, as in this kind of control case, then this ring will expand isotropically. So if we measure just the major divided by the minor axis of the circle, it should be near to 1. Okay. However, if for example, so I'm just trying to explain this in a bit of a cartoon way. So for those who haven't seen this before, it can be a little bit tricky to understand. So our hypothesis is that if you have, for example, a random mesh of basement membrane, which we know is kind of like this kind of meshwork of collagen fibers, you do this ring ablation, and then you look at the stress relaxation by analyzing the shape of the outer ring, but also in the ring if you want to, but the outer ring is less noisy. And so if there is stress, for example, we apply stress by stretching it, and we hypothesize that this induces the fibers to align as you would in a network. We do the same circular ablation, and we should be able to measure this increase in stress that we've applied along the x-axis because the tension will be higher on the long x-axis, and this should be visible by this ring analysis. Okay, so the outer ring will now deform into an ellipse with a longer, major axis along the axis of stretch. Okay, this is simply because there's more tension on the x-axis. So basically, this should deflect more than this. Hopefully, you can see my mouse. Yeah, and however, to then measure the stress diminishing over time, we just continue doing this at different time points along the stretch. For example, after 1 hour, 2 hours, 3 hours, 4 hours. And if the stress does relax, then eventually, even though we maintain the stretch or the strain, the stress should be gone, and we should be able to measure that relaxation of the stress by this circle or this ring going back to the original isotropic deformation. Okay? So hopefully that makes sense. So just to show you in practice what happens to the results. So Nargis is the incredible postdoc who did all this work. So she, for example, did the controls where we anchor just the wing disc in the dish and we cut the apical adherence junctions. And as we predicted, you basically get this isotropic deformation. And so what she does is to wait until the deformation has stabilized. So this is the track over time. So this would be the major axis of the outer ring, okay, changing over time against the minor axis of the outer ring over time. So if it's a circle, then of course it's basically the same. And then she divides the major by minor at the plateau. Okay. And this is if she stretches it, you'll see as we predicted, after let's say 20 minutes of stretch or straight after stretch, then you see this major axis basically deforming much more than the minor axis. Okay. And then if you plot the ratio of major divided minor at the plateau, you should be able to see this. So this would be the control anchor, and this would be the plateau curve. Okay. And so then if she does this for different discs that have been stretched for different times at different time points after stretch, so one caveat with this laser ablation is once you've ablated it, you can't use the same disk again, okay? So these are average points, of course, of many data points where, for example, this wing disk was ablated at 90 minutes, this after, 3 hours, 4 hours, and so on. And you can see, as we said before, that if you stretch a wing disk, the stress is basically maintained up to at least 3 hours, and by 4 hours, it starts to drop, okay? And so it's a very long sustaining of this stress in the tissue after stretch. And so remember, our hypothesis is that actually this stress is maintained because of the basement membrane. If you don't have the basement membrane, what would happen to the stress relaxation time? Would it relax faster? And so what we did is to do this exact set of experiments, but actually remove the basement membrane with collagenase. And what we find is that actually the initial tension goes up very high as the control, but the stress relaxes much faster. After about, between basically 2 hours or so, the stress has already come back to the original stress. And so what this shows is that if you don't have a basement membrane, you have a much shorter mechanical memory, as we call it, and this we think therefore shows that basement membrane is really helping the tissue maintain this long ability to buffer forces and fluctuations in the animal, and of in the tissue of the animal. Okay. And so, but of course, this means that, the basement membrane itself has to be under stress. So just to be clear, what we do here is we ablate the apical surface, the top apical surface of the cells, and the basement membrane, remember, as I said, is really quite basally underneath the cells. And so is the basement membrane itself also under stress. So what we can do is similar experiments, but cutting just the basement membrane. And this is very tricky because the basement membrane we're talking about, a few hundred nanometers thin. And so trying to cut this, which is, thinner than the wavelength of light that we're using is very hard. But luckily, Nagas has been able to do that to assess the tension in the basement membrane itself. And you can see the basement membrane also deforms under the cut, so showing there is tension in the system. But also very cool is sometimes you'll accidentally not cut all the microfibers. And so here you can barely see the fiber, but actually you can see the fiber is bearing tension in the basement membrane. And so this shows that the basement membrane itself does indeed have tension. And so what we've then done is to actually look at how does the basement membrane remodel to, first of all, bear this tension, but also relax the tension. And does that occur at the same time scale as the cell relaxation? And so here, this is live super resolution imaging of the basement membrane, and you can hopefully see that you actually polarize the fibers as you stretch the basement membrane, and you can quantify this polarization, for example, using the Matic order. And we see that essentially after the basement membrane stretch, the polarity is slowly lost as the basement membrane remodels itself, but this remodeling occurs very slowly over the course of 3-4 hours along the same line as the cells relaxing the stress. So this for us shows that essentially the basement membrane is what's allowing the stress to be sustained. When it's stretched, it's polarized, but then it slowly remodels its polarity, relaxing the stress to maintain the stress in the tissue for longer, which would be useful if you want to buffer forces. And we show that if you don't have if you have cells without a basement membrane, then this memory is much shorter. And this, of course, is probably problematic if you're, seeing these fluctuations. So in the last couple of minutes, I just want to show you how this is relevant in vivo. So this is all in vitro, which is great, but we can't look at the long-term consequences of the wing disc developing, right? We can't put it back into animal to look at what happens. And so this is just to show you a larvae as it's developing. Actually, all the organs inside the larvae are constantly moving. Okay, so this is why trying to buffer these forces is very relevant. Otherwise, you'll have deformed wings hatching every time. But as I said, if you focus on, for example, the wing discs, you'll see they undergo fluctuating forces, but every wing, adult wing, hatches out identical. So how do you actually buffer these, right? And so what we've been trying to do to understand is to try to break this force in vivo now. And so this is a normal wing, but we've been making channels where we make these larvae crawl through these very tight constrictions every few millimeters. And as a result, every time the larvae crawls through constrictions, the internal organs, such as the wing disc, get stretched, sometimes more than 100% in this, what we call in vivo stretching devices. And so what we've done is to scale this up, and we can have hundreds of these PDMS-based channels where the larvae are doing the larval Olympics, we call them. And as a result, we can track every single larvae's movement, their basically how active they are, and then actually give each larvae a unique ID and look at their phenotypes in adults when they hatch. And the power of fly genetics is that we get phenotypes in organs, for example, the wing here, that look like classical genetic phenotypes, but we've not done anything to the genes. This is purely mechanical perturbations, but they reflect or almost mimic genetic phenotypes. And so now we want to use this assay to understand, for example, which genes is it because some certain pathways are the most sensitive to these forces that we're applying externally. That's why these pathways are breaking and we get these genetic pathway phenotypes. But also, so we can do modifier screening. So, for example, we can apply sensitized genetic conditions to these mechanical assays and look at synthetic lethality essentially. Okay. And so this requires a lot of work. So we've been actually trying to use this assay in summer schools where undergraduate students can actually work on this assay. And we've had 30 students last year, for example, doing these assays. And I think this could be a really interesting way forward, before we get robots working on these channels, is to be able to increase our numbers, but also teach the students genetics in action and mechanics in action. Um, and so conceptually, I think what I want to end on is this idea that actually classically we study how genes, for example, if this is a gene-gene interaction or protein interaction, if you break that, the whole system collapses, right? But of course, we can also break a system externally by applying external force. So these are quite extreme cases, but of course these lead to diseases. But I think what we're trying to understand is what if the initial trigger is an external environmental factor, UV rays, mechanical forces, whatever. And this actually triggers certain internal links, weak spots to break, and then your system collapses. Okay. And so I think the key is then to ask which are the most sensitive or vulnerable interactions or genes in our system, in our bodies, and is that different for each of us? And I think we can do this potentially in our assay system, where we can combine genetics with mechanics and to do these in vivo modifier screens. And so I think with that, I'd like to end by thanking everyone in my lab, past and present. I've tried to mention some of their names and pictures as I went along. It's mainly the work today of Nargis and Julia that I talked about. But this is the rest of the team and the funding. I'd like to thank all the sources of funding and thank you for your attention.
Let's thank Yanlan for this really wonderful talk. I also see Zonia raised a question in the chat. Zonia, would you like to just ask a question?
Yeah, sure. Yeah, this is really, really fascinating. Um, and so I was wondering, particularly with what you were talking about at the very end, um, in terms of, mimicking genetic phenotypes with mechanical perturbation, is there
Hmm.
any evidence that some of the mechanical stresses can sort of retroactively affect gene expression?
Hmm. Hmm.
Is there any evidence
Yeah.
for that yet?
So in our system we haven't done, for example, single-cell RNA-seq yet, but in other
Mhm.
systems, absolutely. I mean, there's lots
Right,
of data
right,
suggesting mechanical forces affect gene expression, and there's many mechanical sensitive pathways and so on and so forth.
right.
Yes. But
Mhm.
we need to do that in our system. Yes.
Yeah. Yeah, thanks. Yeah, it'll be fascinating to see what you find.
Yes. I
Yeah,
mean, we're just trying to basically tune the assay so because the system is so robust, the phenotypes are very low penetrance. So we want to increase that penetrance a bit more before we go to single-cell RNA-seq. Otherwise, it'll be too
right.
noisy, I think. Yeah.
Yeah. Okay, thank you.